Hi everyone, in this video we're going to continue our discussion on cell structure and function by describing in detail the cell membrane. So the cell membrane is alternatively referred to as the plasma membrane or plasma lemma. And one of the fun things about studying physiology is that there are oftentimes several different words that can be used to describe the same structure. And so the suffix lemma is what refers to the membrane, and so there will be other membranes that we'll refer to that can still be called plasma membrane, but will be specific to other structures. So for example, we'll say sarcolemma or neurolemma, and in those cases we're referring to the cell membrane for that particular type of cell. So it's extremely common to see the word lemma, but I will mostly refer to it as cell membrane. There are several important functions of the cell membrane. The first is physical isolation. The conditions inside and outside of the cell are very different. We mentioned that already. They have different ion concentrations, and there's a slightly different pH. So there are things that are different about the cell. And so it's very, very important that what's inside of the cell stays, that environment stays stable. And if that environment is exposed to the outer conditions, then it can disrupt the homeostasis of that particular cell. Second, regulation of exchange with the environment. So the cell membrane is responsible for controlling the entry of things like ions, nutrients such as glucose, elimination of waste products such as carbon dioxide, and release of certain types of secretions. The cell membrane is also very sensitive to the environment. It's the first part of the cell that's going to be affected by changes in pH, composition of the fluids in the extracellular space, concentration of specific ions, things like that. And so it also contains receptors that can receive information. So if anything is changing externally to the cell membrane is going to be the thing that's able to identify that change and then either prepare the cell for it or modify itself in some way so that it can protect from any kind of negative change. Finally, the cell membrane is responsible for structural support. So cells make special connections with each other. We won't cover this specifically until we start talking about tissues, but these connections allow for stability, allow cells to remain in close contact, allow cells to communicate with each other. So these this one will kind of put on hold, but it's no less important than the other functions. So let's talk a little bit about the structure. So the organic molecules that are utilized in order to build the cell membrane are lipids, proteins, and carbohydrates. And so the lipids and proteins are both very, very abundant, and the carbohydrates a little less than the lipids and the proteins. So we'll start by describing the lipids. So we mentioned in one of the videos in chemistry that the phospholipid is what makes up the cell membrane. And if you remember, the phospholipid has a polar head and it has a nonpolar tail. And the nonpolar tail is two fatty acids, one of which has a double bond. So one of the legs is bent and the other one is straight. For the purpose of our drawing, we will draw them both straight most of the time just for ease of drawing, but doesn't make it doesn't mean that the structure has changed. So what we do to build the membrane is we actually string a bunch of these together, and we can make sort of a double layer. So the polar heads orient away from each other and the nonpolar tails orient towards each other. And the reason for this is that we're going to place this structure in water. So the polar heads want to associate with water, and the nonpolar tails do not want to associate with water, and so sort of naturally they will orient themselves. They'll move in such a way that the polar heads are all sort of facing the water on either side of the cell, inside and outside, and then the tails are facing towards each other and are protected by the polar heads from that water. So as we draw our drawing of the cell membrane, we'll oftentimes descriptively identify which is the inside of the cell and which is the outside of the cell. And so sometimes we'll draw it in a circle, but most often when we're talking about something that's happening at the cell membrane, we'll sort of draw it like this. So we'll label the outside of the cell and the inside of the cell. And just remember that the inside of the cell contains intracellular fluid, which is of a certain composition, and the outside contains interstitial fluid, which is of a different composition. So the polar heads, remember, are hydrophilic, and the nonpolar tails are hydrophobic. And together, this construction is called the phospholipid bilayer. So two layers, bilayer, of phospholipids oriented in such a way that the hydrophilic portions face the inside and outside of the cell, face the water, and the hydrophobic portion is shielded. This type of membrane is also referred to as semi-permeable. It's a semi-permeable membrane, meaning that it's a selective barrier. So it has the ability to isolate the intracellular fluid from the extracellular fluid and decide which molecules are able to enter the cell and which ones are not. So without talking about the proteins that we'll find in the cell, generally speaking, hydrophobic molecules can pass through the membrane easily, but hydrophilic molecules cannot. So if you are small and lipid soluble, and this is what we'll say most of the time, small and lipid soluble, you can diffuse right through the membrane easily. And if you are not, if you are charged, if you're polar, if you're big, then it will be very difficult for you to move through the membrane, and we'll have to find some other ways to get those types of molecules in if we need them to come in, or out if we need them to go out. But otherwise, the cell membrane is able to specifically decide if any particular molecule has a key to enter. One other type of lipid that's worth mentioning here besides the phospholipid is the steroid. So cholesterol is oftentimes a component of cell membranes. We'll just draw that in purple. By far less important than the phospholipid for our purposes, but in terms of maintenance of the cell membrane stability, cholesterol is one of the molecules that contributes to that function. All right, so now let's talk a little bit about the membrane proteins. So a membrane protein could be what we call integral, meaning embedded in the membrane in some way. And if it's embedded in the membrane such that the protein is exposed on both the inside and outside of the cell, so it extends the entire span of the membrane, we call it transmembrane. And then there are some proteins that are considered integral because they're somehow embedded in the membrane, but they're not spanning the entire membrane. So we can sort of describe those as partially embedded, but we'll still call those integral proteins. Other types of proteins are called peripheral proteins. These are more surface proteins. So the surface proteins will often attach to proteins that are embedded or partially embedded, and the surface proteins are able to detach from the membrane if need be. And we'll actually draw some specific types of proteins that will do this later on. It's involved in processes called signal transduction. So it's kind of like a cell communication type function, and we'll see some of these proteins and see that they can in fact detach from the membrane. So those are peripheral proteins, and they can be on the inside of the cell or on the outside of the cell. So the membrane proteins have 6 major functions, and they're sort of like different types of proteins that accomplish those functions. So what we're going to do now is overview the 6 types of membrane proteins, and then later when we talk about membrane transport, we will sort of identify which proteins are responsible for which types of membrane transport. So the first type of protein that we'll address is the channel protein. So we'll sort of just draw our cell membrane, and then we'll draw our protein embedded right into the membrane, and this is an example of a transmembrane protein. And so basically, you know, we mentioned that molecules that are small and lipid soluble have the ability to diffuse right through the membrane. So oxygen is a perfect example of something that's small and lipid soluble. So it can just squeeze in between the phospholipids to move from one side of the membrane to the other, or the reverse. It can go in both directions. But something that is charged, for example, a sodium ion, if it tries to scoot through the membrane, it's going to bounce off. It doesn't have the ability to move through that hydrophobic component of the membrane. So instead, what we do is we put a channel into the membrane. So think of this as just nothing more than a tunnel, and the sodium ion is able to move through the tunnel in order to move into or out of the cell. There are several different types of channel proteins that we'll address later, and sometimes these channel proteins are just open, and sometimes they're guarded, meaning that certain conditions have to be met in order for the channel to sort of open its doors and allow the solutes to move through. Commonly, the things that will move through these types of channels are water, ions, and small solutes. So it still has to meet the small criteria because the channels are very small, but things that are polar or charged are able to move through these types of channels. Next, we have the carrier protein. So let's consider a molecule such as glucose. So glucose is a little bit too big to fit through channel protein. It's not going to work. So we can see that if it tries to move through the channel protein, it's just going to sort of bounce off. But we do want to move glucose into the cells. So we're going to use a different kind of protein called a carrier protein in order to do that. So what happens is a carrier protein is sort of like open on one side and then it can sort of close on that side and open on the other side to sort of move something through. So the glucose molecule will bind to one of the inside pieces of the carrier protein, and the binding is what activates the movement. So it will actually sort of close over the glucose molecule, open on the reverse side. So the glucose will be released into the cell at this point. But actually probably an even easier example is thinking of this as a revolving door. Sometimes in a cartoon drawing, especially when we're drawing a lot of membrane proteins, we'll sort of draw it like this instead, where we see a revolving door. And so you can imagine that a revolving door, when you step into the revolving door and you press on the door to make it move, that is sort of like similar to the carrier protein and the glucose binding, right? So there's contact, and then you push the door, and then the door rotates, and then you hop out on the other side. So the benefit of the carrier protein in comparison to the channel protein is it allows for larger molecules to be able to move one side of the cell membrane to the other, into the cell or out of the cell as needed. Next, let's talk a little bit about enzymes. So enzymes can be embedded in the membrane, and in this example, we see it partially embedded, and they can be facing either the ECF or the ICF. And so enzymes basically are responsible for catalyzing reactions, and that can be on either side of the cell depending on where they're working. As an example, we can use an enzyme to convert ATP to ADP, and we can utilize the energy that's released in that reaction for something else, another process that needs to occur inside of the cell. One thing that's important about enzymes is that enzymes can actually be part of other molecules as well, or connected to other molecules, other proteins that have other functions in the cell membrane. Next is receptor. So receptor is another example of a protein that's going to work cooperatively. So if we draw, again, a channel protein, and this time we'll represent it slightly different, but we'll still say that it's a channel protein in the membrane. The channel protein can have a receptor, so another protein or a piece of protein that is connected to that channel. So we have these special chemicals that will bind to the receptor, and when they bind to the receptor, it can cause, for example, the channel to open. And when the channel opens, whatever wants to pass through the channel, maybe a sodium ion, will be able to move through, but without the binding of that chemical, the channel will be closed and therefore impassable. So the chemical is referred to as a ligand. So it's just a special chemical messenger that we utilize for this purpose. We call it a ligand. And binding, we'll just say, causes activity. And this is actually a concept that requires much expansion, but it will be more useful after we've discussed membrane transport. And so we're going to revisit this very frequently. It's actually also one of the major principles for drug mechanisms. So it's very important. Okay, next we have anchors. So the next two are going to be kind of brief because we're going to talk about them less in this class. But anchors are basically proteins that are partially embedded in the membrane that allow for connection to other structures, such as the cytoskeleton, which is basically the scaffolding inside of the cell that holds its structure together and kind of like keeps its shape. Anchors also allow for the attachment of the cell membrane to other cells. So this can kind of work both ways. We can use it for the inside of the cell and for the cells that would be outside of it. And it can be utilized to stabilize its position relative to other body structures. And lastly, identifiers are basically, I like to think of identifiers as like every cell has its own flag, and it's either waving a flag that is like, hey, I'm a body flag, or it's waving a flag that's like, hey, I'm not a body flag, I'm a disease, I'm a virus, I'm a bacteria, I'm toxic, I'm something that doesn't belong in the body. So the protein, it's usually important for the immune function. So when immune cells are in different body tissues and body fluids, they can pass by a cell and say, oh, yep, they have the flag that belongs to the body, so therefore they're a body cell, they're safe, they're fine. And then conversely, if they find a cell that doesn't belong to the body because it has the wrong flag, then they can be like, oh, you're dangerous, and it will take the steps to mitigate that danger. Okay, so lastly, let's talk just for a moment about carbohydrates. So in relative comparison, there's a lot less to say about carbohydrates, but they form complex molecules with lipids and proteins. So most commonly, they're shown as a composite of carbohydrate and lipid, or carbohydrate and protein. They're usually found on the outer surface of the membrane. And their functions include cell lubrication and adhesion, especially if we're talking about connecting to other cells. They can be a receptor for extracellular materials. And they participate in cell recognition, so sort of in conjunction with the protein that waves the flag. So normally we draw the cell membrane as sort of like a single layer of membrane, but it's important to know that when we think of a cell, we need to think of it in 3D form. It's going to be a 3D structure. Sometimes they'll be cube shaped, sometimes they'll be like a column shaped. There's many shapes for cells, but all of them are three-dimensional, and so therefore I just want to take a second to draw a three-dimensional version of the cell membrane. So it's still cut out, but you can see it sort of from a little bit bigger perspective. And also take note of the fact that the different types of proteins are found in the same cell membrane. So not every protein in every membrane, but a lot of times a lot of the proteins are there. So we can see the presence of channel proteins, we can see the presence of the identifier proteins, we can see enzymes, and note that they're not just, sort of in the plane of the drawing that we normally draw, but that they can be anywhere embedded in the membrane, all the way around the cell. Okay, so that's it for cell membrane structure and a quick overview of its functions. The major function for the cell membrane is membrane transport, how we get stuff in and out of the cell. And it's actually going to take us probably a couple of videos, at least one, but maybe a couple of videos to really talk about all of those topics. So hopefully you can take a little while to let some of these principles sink in. And then when we do talk about membrane transport, that's the one that we're going to revisit over and over and over and over from now until the end of physiology. So hopefully this is a good overview and a good start. So I hope you enjoyed it. Thanks for watching.